Good morning, everybody, and welcome to our last breakaway session for this year's SAAB conference. So session 2 on plant taxonomy, systematics and plant anatomy. This will be a relatively short session. Unfortunately, our second presenter, Mr. Obose, has fallen ill, so we will just move up the last two sessions. So we'll just have 3 presenters for today. So first up is Ms. Natasha Lombard. Natasha, are you ready for your presentation?
Yes, thank you so much.
Excellent. I will just warn you when you have gone through 17 minutes of your presentation and you have 3 minutes left.
Thank you.
Okay, thank you. We look forward to your talk.
Okay, if you can just give me a thumbs up if you can see my screen and hear me. Can you see the screen?
Yes, we can see the screen. You can proceed.
Okay, thank you so much. Hi, everyone. As introduced, I'm Natasha. I'm a PhD student through the University of Johannesburg and SAAB. Now be presenting a part of my PhD study that was published in 2021. So just a quick introduction to the plants that I'm working on. The genus Thesium forms part of the family Xanthulaceae. Plants of the genus are root hemiparasites, meaning that they parasitize on the roots of other plants. So in the first figure here, you can see this dark brown root is the host root, and then this light brown root is the Thesium root. The Thesium root then forms this extension and this historium around the host, and this is where the exchange of nutrients happens. So these plants grow as herbs or as shrubs. They have these narrow and linear or sometimes scale-like leaves and these dry nut-like fruits. It's quite a large genus. It has about 350 species occurring worldwide. The majority of species are found in the Old World, but we have the most species in Africa, and specifically in Southern Africa, we have about 175 species. So Thesium was last revised in 1925 by Dr. Hill in Flora capensis, but these treatments and identification keys are now out of date. Since then, about 50 new species have been described in South Africa alone, and there's now previously unknown intraspecific variation that has been observed. So as a result of this, species delineations are unclear for many of the species, and they need to be revised. So Thesium has therefore been listed as a high priority for taxonomic research in South Africa. So this group of species that we revised, we actually came across them when doing curation in the herbarium, and we realized that it was quite difficult to place the specimens with the species. So we then decided to go into more detail and to study this. And we actually found these 8 taxa that formed part of this complex, of which 3 were listed as data deficient due to taxonomic reasons by the IUCN. So this complex, it's part of the monophyletic subgenus Freesia, according to the molecular phylogeny done in 2020. However, we do have incomplete information on monophyletic groupings at species levels within the subgenus. And in the study, the 2020 study, only 3 of the 8 taxa were included, and there was some incongruence between nuclear and plastid trees. So we therefore opted to use a morphological species concept combined then with geographical distributions in the study. Plants of this group share these few characteristics. So they have these inflorescences that are indeterminate and that are spike-like. We say spike-like because the oldest flower is not always at the bottom. Then they have these very reduced and scale-like leaves. these hairs, I don't know if you can see there, but these small hairs connecting the stamens to the side of the flower, twisted placental columns, there you can see the loop, and they're normally inconspicuous and unarmed. They grow with these herbaceous and suffrutescent habits or growth forms. So the aims of this study was then to reassess and confirm the taxon concepts within the complex, to provide a full taxonomic treatment for each taxon, as well as an up-to-date identification key. And we used standard methods, so we actually did quite a lot of field work across South Africa between 2016 and 2020, where we managed to collect and photograph 6 of the 8 taxa. We also studied the taxonomic literature and the type specimens, and looked at morphological characters of about 360 specimens from 8 herbaria worldwide. So then we gathered data for 5 vegetative and 13 reproductive characters in the end, and formulated species concepts and reviewed the nomenclature. We compiled an identification key and georeferenced herbarium specimens, which we then used to make distribution maps for each species to show the geographical distribution. And we also determined preliminary conservation statuses using IUCN guidelines. So going into the results, some of the interesting things that we found. Firstly, in terms of Thesium jungium, it had 3 varieties. The first being Thesium jungium bar plantagenium, which is characterized by these very compact inflorescences that resemble those of the genus Plantago and Plantagenaceae. And then you can see here the specimens that we collected in the field with these very compact inflorescences. And then you have Vardiunkium, which is a sort of a more lax plant with lax inflorescences. And at first, it seemed very clear the difference between these 2 varieties. But as we looked at more specimens, the distinction became less clear. So here on the one side, you have the typical Var plantagenium specimens that occur sort of in this range. And then on this other end, you have the typical Vardiunkium specimens that occur here. But in between, you find specimens, this one sort of has a lax habit, but more or less compact inflorescences, while this one's habit maybe more resembles that one. So the takeaway here is that it became difficult and actually impossible to distinguish these 2 varieties from one another. And we actually realized that plants that were quite compact and with compact inflorescences were the ones that actually grow in the mountain areas, in the mountainous areas here. So it's probably more an artifact of the environment. And then the third variety, Var mammuosum, mammuosum meaning breast, referring to these protrusions that you see here on the flower lobes. This variety was actually described from only 1 specimen. This is a flower from the type specimen. So you can see the protrusions are quite prominent. But then you find other plants where the protrusions are there, but not as prominent. And then you find other plants where the protrusions are completely absent. So it's the same story, and it became very difficult to draw distinction for the boundaries of this taxon. We then looked at any other characteristics that might be able to distinguish this taxon, but these were the 5 specimens that we found with protrusions. There is no other characteristics that can group them together. As you can see, they also show this distinctive lax habit in the type, all the way down to this distinctive compact habit. So then there was no grounds to retain any of the varieties of Jungium. And then another interesting thing that we came across while I was working in the Salmarshonland Herbarium in Grahamstown was the specimen. It was something more or less between Thesium lisa-mariae and Thesium juncium, or maybe Thesium flexiosum. But it had these very distinctive dark, almost black bracts. So we only had one specimen of this species at the time. We went to iNaturalist, to look in the same area, and we actually found this collection made by Nicola van Berkel. And lo and behold, it's the same plant. It has these same, there you can see very clearly these dark bracts. And we confirmed that this was a species new to science, and we then named it Thesium atratum after the dark bracts. So just a summary of the nomenclatural changes that we made to this complex. We then described Thesium atratum as a new species. And then in Thesium juncium, the varieties were all sunk under Thesium juncium, so that 7 species are now accepted. And then how to distinguish each of the species. I'll just go through their diagnostic characters. The Thesium flexiosum is quite distinctive because it has these very compact and sort of curved inflorescences, and it grows quite robust. As you can see, the inflorescences are upturned all around. And then it has these distinctive hooded flower lobes, so the hoods are quite prominent, and this very sparse beard. We call it a beard because these hairs resemble a beard. And then it also has these bracts with these broad cartilaginous margins. And then there you can see the distribution. So Thesium flexiosum is very similar to Thesium paranochioides. You can see it also has those hooded flower lobes and that very sparse beard, these same bract margins. But at the time of publishing, we had only studied this species from specimens, so we had not yet found it in the field. So we only knew it from the type collection here in Swellendam, and then a few other collections. But this locality, because it was so far removed, we were not sure that it was correct. But then at the end of last year, lo and behold, after the study was already published, we found this plant in the field, which then corresponds perfectly with our concept of it. We found it in this area, so we can then confirm this is a separate species, this locality is correct, and it probably does occur in between, in this area in between. And it can be distinguished from the previous species, Thesium flexiosum, in these straight inflorescences. So that was quite exciting to find this species also. The Thesium juncium has the same bracts as the previous 2 species, but it's completely different in that it has this dense beard on the inside of the flowers. And it's actually the only species in this complex that has these sterile bracts, this sterile outgrowth in the inflorescence. You don't find this in any of the other species. And then you can see the distribution there. So Thesium juncium might be confused with Thesium natalense. It also has quite a dense beard, and in its habit you might also be confused, but the bracts of this species do not have those broad cartilaginous margins, so that's an easy way to distinguish them. And the habit, it normally only has a few stems at ground level, normally unbranched for the first 2/3, and then branched in the upper part. And it grows quite tall, up to about 70 centimeters tall, and it's concentrated in KZN. It's more likely to be confused with Thesium scirpioides, which their distributions overlap. They also have very similar flowers as the previous 2 species, and also very similar bracts as Thesium natalense. However, this plant is mostly unbranched or branched from the base, and it has multiple stems at ground level. The inflorescences are also, the flowers are much more spaced in the inflorescences, whereas in Thesium natalense, the inflorescences are more compact. And then the last two species, Thesia matratum, as we already saw, it has these dark bracts, and then the flowers have this flat disc at the base of the lobe, which is absent in all of the previous species. And the flower lobes are glabrous, so no beard. It's very similar to Thesium lisa-mariae in that it also has this disc in the flower and glabrous or erose lobe margins. But as you can see here, the bracts are not dark. So the bracts are not blackened, and also the inflorescences are much more elongate than in Thesia matratum, where they are much more compact. So these differences are summarized in this table, and here I've just circled the main diagnostic characters for each of the species, so that one can just at a glance see the difference, where the differences between species lie. And then this is the identification key that we produced, which I always feel is one of the most important parts of the work that we do, because this is what the end user will be using to identify their plants. And then looking at the conservation statuses, preliminary conservation statuses that we determined, Thesia matratum will likely be listed as endangered because it's only known from a few localities, but many of those localities are in nature conservation areas. Then Thesium flexiosum, Thesium lisa-mariae, and Thesium scirpioides are very common plants, and they are then listed as least concern and remain listed as least concern. As I said before, Thesium juncium, varmamosum, and varplanticinium were listed as data deficient due to taxonomic problems. This is then now resolved, and the same for Thesium lisa-mariae. However, in the case of Thesium lisa-mariae, we still have insufficient data. So the taxonomy is resolved, but we only know it from 2 or 3 localities and specimens, in fact. So we have very little data on it, but it will probably also be listed as endangered. And then with Thesium paranithioides, this plant, I think, is much less common than previously thought. The majority of the specimens were wrongly identified. So I think it will also be listed as endangered. And this will, of course, be confirmed by the Red List team at Sunbeam. So in conclusion, the previous species delineations within the Thesium scirpioides complex were quite unclear and needed to be revised. And then we identified the initial 8 taxa as part of the complex, of which 3 were then data deficient due to taxonomic reasons. And this has now been resolved. We then looked at 5 vegetative and 13 reproductive morphological characters, and also the natural distributions of these species. And we actually saw that some of the morphological characters showed a continuum of variation. And as a result, Thesium juncium, varmamosum, and varplanticinium could not be retained as distinct taxa, and they were then synonymized under Thesium juncium. So currently we have 7 taxa species that are now recognized. Now I would just like to thank the following organizations and also all of the people involved with these organizations for supporting and funding this study. Thank you so much.
Thank you for an excellent presentation, Natasha. That was really interesting, and I think your study really demonstrates how important fieldwork is in taxonomic research. So we have 2 questions. The first one is from Anina Kutsia, who asks, do Thesia mostly reproduce sexually through pollination or asexually? Anything known about the pollinators?
Okay, so for the first question, I think it's both. Thesiums are remarkably well adapted to survival. So when you go into the field, when you do field work, normally we always make a joke that when you put your foot out of the car, you will step on a thesium. That is how easy it is to find them. And even when nothing else grows in the field, there will be a thesium and it will be in flower. So in terms of that, they are very adapted survival. And some of the plants do make rhizomes. So they will reproduce vegetatively through rhizomes below ground, which will then form more plants, but they also do produce seed. And then the second question was the pollinators. There are no official reports on the pollinators, but just from field work, what I've seen, we've seen bees, we've seen flies, we've seen ants, we've seen wasps. So we've seen a wide variety of insects that probably contribute to pollination.
Fascinating. Glynis says, thanks, Natasha, an excellent presentation with very clear images. I know you applied a morphological species concept, but wondered if varieties of Thesium junceum group together in the phylogenies.
So we don't have this information. Unfortunately, in an ideal world, we would love to have multiple sequences for each of the taxa, but there were only 3 species included in the phylogeny, the 2020 phylogeny, Thesium flexiosum, Paronychioides, and Lisa mariae. So we unfortunately do not have this data. It would, of course, be ideal to have it. And in the future, it would be interesting to see if it does corroborate our results.
Yes, it would definitely be useful to do more detailed or molecular studies on it. I think it would be interesting. And then a final question from Conrad Geldnys. Is the taxonomic investigation of Thesium ongoing for other regions of the country, such as the succulent Karoo?
Yes, so currently the entire genus is being revised by me. It's a big undertaking. It will take some time. So we're taking a systematic approach where we revise smaller groups of species, because there's 350 species, 170 of those in Southern Africa. So yes, it's ongoing, and we will continue publishing these revisions. I think this is now the 3rd revision that we have published. There's also a group from UCT with Prof. Moasia, working on more of the Cape species. So yes, definitely we'll be doing that.
Exciting work. Thank you very much, Natasha. There's just a final comment by Janine Victor. This is valuable research that will enable conservation and management of a previously poorly known genus. Indeed. Thank you very much, and good luck for the rest of your research.
Thank you so much.
Right, so as mentioned before, unfortunately, Mr. Obosi has fallen ill and is unable to join us today. So we are going to move ahead to our next presenter, Mr. Goitsione Sedimu. Goitsione, are you up and able to share your presentation with us?
You can just activate your camera and microphone. You're currently backstage, so just on the bottom of your toolbar, put your microphone and your camera on. Then we can see you, and our technical team will share your slides.
Good morning.
Good morning, Goitsione, and welcome. So Goitsione will be presenting his project on the taxonomic revision and molecular phylogenetic analysis of Namibia in the Free State. And we can see the presentation. So Goitsione, you can go ahead and then just indicate to our technical team when they need to progress your slide. Goitsione, just make sure your microphone is switched on so we can hear you.
Can you see the first slide? Koetsione? It seems that you have poor connectivity. If you can just
Part
to
of
the
the Menoridae
slides.
tribe, which is the earliest branch ranking. 12 Nemesia species occur in the Free State province, and those are the species that are listed on the table attached.
Next slide, please. Addition,
Nemesia
was named as one of
to general data in need of taxonomic revision by Sunday in 2015 on a list compiled based on factors including time since last revision, level of endemism, collection effort, proportion of taxa included in revisions, and species identification confidence. And interestingly, the last time the Nemesia genus was revised was over 100 years ago by Hien in 1904. And the revision included 48 species,
of which 8 occur in the Free State. And not only have new species been described after, but some nomenclatural changes have been made to some of described by Hien. For example, Nemesia flanaganae is name of Nemesia albiflora. And the only published molecular phylogeny of Nemesia is the one constructed by Dodson et al. Using a nucleotide sequences of nuclear ITS, the ATS, and the chloroplast tRNA spacer regions. The study included 23 Nemesia species, and only 4 of them were species occurring in a Free State. So the Free State Nemesia species are the ones that are indicated with the maroon boxes on the diagram from Dodson et al., which is attached on the right side of the slide. Next slide, please. Okay. So this shows that there's a huge gap in our knowledge about Nemesia species and their evolutionary relationships. It is therefore important for us to expand the phylogeny of Nemesia and to revise the taxonomy of the genus in order for us to fill these gaps. So with this project, we aimed to complete a systematic analysis of Nemesia species occurring in a Free State. And this was achieved by revising and correcting the taxonomy, the nomenclature, typification, and the morphological description of the species. And we also included as many Free State Nemesia species to the Nemesia phylogeny as possible through the analysis of the 3 non-coding gene regions. Next slide, please. So for the taxonomic revision, we studied all the original species descriptions, the successive revisions and type specimens with the help of online sources like JSTOR and the Biodiversity Heritage Library. We also studied herbarium specimens to test the species delimitations using specimens from the Geopods and the National Herbarium. The Q plant glossary was also used to find the correct terms to describe the specimens. And a nomenclatural evaluation was also conducted following the rules of the ICN. Next slide, please. When it comes to adding as many Free State Nemesia species to the phylogeny, we added as many Free State Nemesia species to the unpublished phylogeny, which was constructed by Hanley for her master's using the same gene regions that she used and the same gene regions used by Dodson et al. In 2008. So this was done in 4 steps, with the first step being DNA extraction, where total genomic DNA was extracted from leaves of herbarium plant materials using a modified C-type method described by Doyle and Doyle in 1987. The specimens used in the study are the ones that are indicated on the grade table on the left side of this slide. The second step was PCR, where the 3 non-coding gene regions were PCR amplified. And the PCR was performed using the primers that I mentioned on the maroon table that is on the right side of this slide. Next slide, please. So our third step was DNA sequencing, where the DNA sequencing reactions were conducted using the Big Dye Terminator Kit version 3.1 according to the manufacturer's specifications. The same primers used in PCR amplification were used in the respective reactions for both the forward and reverse reactions. And our last step, which is phylogenetic analysis, is where the sequences were edited using BioEdit and Chromas Pro. And they were then blasted online using the BLAST website. And they were aligned using MAFT, and the consensus sequences were generated using BioEdit. Next slide, please. Okay. Oh, first, the POP was used to perform the maximum parsimony analysis, and Mr. Bayes was used to conduct the Bayesian inference analysis, and the J-model test was also performed to determine the model of evolution. Okay, and now, okay, once more. Okay, stop. So, how do we identify Free State Nemesia species? Free State Nemesia species are annual or perennial herbs, or sometimes fruitexes. They have leaves that are simple, opposite, and not stipulate. Their leaf blades are herbaceous, and their inflorescences are slender terminal racemes with bracts that subtend the pedicel and have an entire margin. The perianth of this species is divided into a calyx and corolla, with the calyx being free, actinomorphic, and 5 mirrors, with an entire margin, and the corolla being fused and zygomorphic with a 4-lobed upper lip. Their stamens are uniseriate, 4 mirrors, and didynamous, with anthers that are dorsifixed and filaments that are tubular. And their gynoecium is syncarpous, with a superior ovary that is flattened and orbicular, and they have 1 style per pistil. And lastly, their fruits are capsules with ovoid seeds that have 2 wings on the side. Next slide, please. Okay. Now let's take a look at the phylogeny of Nemesia. The phylogeny is divided into 2 big clades, namely clade A and clade B, with clade A being relatively more resolved than clade B. And interestingly, Nemesia species occurring in a free state all occur in clade A of our phylogeny, and they include all the species that are indicated by the maroon boxes. Next slide, please. So as we take a closer look into clade A, we can clearly see our free state Nemesia species in those maroon boxes. And this species falling within the same clade indicates that there's a close evolutionary relationship between them. And this might explain why some of them are so morphologically similar. Next slide, please. Our phylogeny helped us solve some of the species circumscription problems that we came across. And one of the examples is the Nemesia novirica and Nemesia pubescens problem. So it was a big challenge to differentiate between Nemesia novirica and Nemesia pubescens specimens. And this is because apart from both species having white flowers, they both had pubescent surfaces, and their nectar spurs were broad or saccate with a tip that points downwards. And lastly, their fruits all had blunt tips. Next slide, please. These species also have an almost similar distribution in the Free State province. So as you can see here on the distribution maps, they're both found on the western side of the Free State province. Next slide, please. Okay. So, however, our phylogeny clearly shows that these are two different species as they are found in two closely related sister clades. And this shows us how important molecular data is when conducting a taxonomic revision. Next slide, please. So another problem that we had was the Nemesia fruitans and Nemesia fruticans problem. So Nemesia fruitans has been internationally accepted as a synonym of Nemesia fruticans, and it is listed as Nemesia fruticans in scientific websites including Plants of the World, Catalogue of Life, and IPMI. However, in South Africa, Nemesia fruitans is still recognized as a species on its own, and it has been listed as a species in Retief and Meyer 2017 and Sunbee's plant list. Therefore, morphological and molecular data of specimens of the two species were compared as an attempt to solve the classification problems. We found that Nemesia fruticans had nectar spurs that are tubular and slender, while Nemesia fruitans had nectar spurs that are tubular with a broad base. And in addition, specimens of Nemesia fruticans had fruits with apices that are emarginate, while Nemesia fruitans had fruits with apices that are truncate. And the evolutionary relationship of this species is not clear because, as you can see on the phylogenetic tree, their specimens occur on 4 different parts of the phylogeny. And this might mean that the specimens were misidentified and they're actually 4 different species, but we don't know that yet. So therefore, more morphological and micromorphological studies must be conducted to solve the classification problem of this species. And more specimens should be collected and added to the phylogeny to see where they will be on the phylogeny relative to the ones that we already have. Next slide, please. Following the rules of the ICN, especially the one that states that for a name to be valid, a holotype must be designated. Many typification problems of Nemesia species in the Free State were solved. For example, no holotype was designated for species including Nemesia albiflora, Nemesia fruticans, and Nemesia pubescens. So we corrected this by designating a single lectotype for each species. Next slide, please. A good example of a typification problem being solved is in the case of Nemesia flanaganae, which is a synonym of Nemesia albiflora. In this case, Hilliard, in her taxonomic revision, listed Flanagan 1617 as a lectotype of the species. However, she did not specify at which herbarium the specimen is stored, since Flanagan collected many specimens with the same collector's number, which is Flanagan 1617, and they are stored in different herbaria. So to solve this, we designated one Flanagan 1617 specimen as the lectotype of Nemesia flanaganae, and that is the Pretoria specimen. We also found other Flanagan 1617 specimens, and we listed them as isolectotypes of Flanagan of Nemesia flanaganae. So in conclusion, Nemesia species occurring in a Free State are closely related, and this is reflected in their morphological similarities and their molecular data. And with Nemesia being a priority genera for taxonomic revision, a better representation of Free State Nemesia in the phylogeny of the genus will play a significant role in filling the gap in our knowledge about the evolution of Nemesia species and their relationships. And also, The correction and verification of the typification and nomenclature of this species will also help us avoid mistakes in future studies of this species. Next slide, please. So these are my references. Next slide, please. I would like to thank my supervisors. Sorry, please go back.
For their guidance and support throughout the project.
And I would also like to thank Emma and Hanley for providing me with the distribution maps and the phylogeny of Nemesia, respectively, as this helped me interpret my studies and my results better. And lastly, I would like to send a special thanks to my family for their love and support throughout my project. So that is all for me. Thank you.
Thank you, Gwetsione. Very interesting talk. We have a couple of questions. The first question is from Glennis. So she says, hi, Gwetsione, thank you. I'm interested in your choice of ITS primers. Are they specific for Scrophulariaceae or better than the White et al. Primers? Or why did you use these specific primers?
We used these primers. Okay, that's fine. We use these primers because for the past studies of the Nemesia, the past studies that were constructing the phylogeny of Nemesia used the same primers and the same non-coding regions. So for our
Gwetsione, are you still there? Okay, unfortunately, it seems that we have lost connectivity with Gwetsione. All right, but Gwetsione, are you back?
Yeah.
Maybe just repeat your answer. We lost you there for a moment. Okay, it seems that we have lost our connection, but luckily it held for most of the presentation. So then there's also just a comment for honors level. This is an excellent work. Well done, Gwetsione. So well done and good luck for the rest of your studies and everything you're planning for the future. Then last up for today's session is Hanley Robler. So Hanley, are you available and ready to give your presentation? All right, Hanley, welcome. Are you ready to share your presentation? Let's see if you can share your screen. So while Hanley is sharing, I will just say that Hanley is presenting her talk on nectar spur development in Indonesia. So another Indonesia talk. Hanley, I will let you know when you're on 17 minutes and you have 3 minutes to conclude.
Okay, thank you. Can you see my screen?
Yeah, you can go ahead.
Okay, so today I'm going to tell you more about development in Nemesia, a genus in the Scrophulariaceae family. Just a brief introduction to Nemesia. The flowers show great diversity in size, shape, and color among the different species. And the single floral spur differs in curvature, orientation, and shape among species. The spur may contain nectar as a reward for visiting pollinators. The floral spur is a classic example of a key innovation, which is a newly evolved trait linked to increased diversification. Understanding the development of a key innovation is critical in understanding the evolution of diversity in flowering plants. Spurs have ecological and evolutionary importance as they influence the pollinated type, pollination efficiency, and reproductive success of the plant. Floral spurs evolved independently in related flowering plant families, and because of that, differences in the origin and basis of spur development occur. The development of a spur is due to cell division and the process contribute to variation in spur shape. For example, spur length variation among species is due to cell expansion, with the shorter spurs having shorter cells compared to the longer spurs, which have more elongated cells. Another example can be seen in Indonesia, where spur length variation occur among two sister species, with the long spur having 3 times the number of cells compared to the shorter spur, while the cell size were approximately similar, showing that the differences were due to cell number only. Now, the aim of my study was to determine the developmental basis in Indonesia by using a comparative approach to investigate if variation in cell size and or cell number contribute to spur variation among species. So my materials and methods, we started by selecting 3 study species which show spur morphological variation. We have Nemesia barbata with a conical spur, Nemesia macrocarpa with a narrow, long tubular spur, and Nemesia Thromosa with a very broad saccade spur. In the measurements we did to compare the species, we did light microscopy and scanning electron microscopy, or SEM, using a dyno-light microscope, which looked like this. We measured the daily spur growth over spur length and width, and then using the SEM, we captured SEM images of the external nectar spur epidermal cells to compare cell number over spur length and width and cell size among the 3 species. So for light microscopy, we measured spur length and width daily as soon as the sepals opened until enthesis, because that's when spur development discontinued. And we used R to construct growth curves and do the ANOVA test to test the significance of differences. And then we also compared the growth rate and growth duration of the 3 species. So to measure spur length, we first had to determine a consistent point to measure spur length from, and that was the spur base we selected, which is indicated by the arrow for each of the species. That is where the upper and lower corolla lips meet, forming the spur base, or also the entrance of the spur. And then from the spur base, we measured the spur length from the spur base to the tip of the spur for the 3 species. And then spur width was measured halfway between the base of the spur and the tip of the spur. We did the measurement in this way to ensure consistency of measurements within and among species. Then after followed the spur growth, we could determine 5 developmental stages. So for Nemesia barbata and Nemesia macrocarpa, spur growth continues for 9 days, while for Nemesia Thromosa, it continued for 13 days. So for barbata and macrocarpa, we selected a stage on every 2nd day, and for Nemesia macrocarpa, a stage was selected on every 3rd day of spur growth. So stage 1 looked as follows. That was when the sepals opened and the spur became visible, and we could start measurements of spur length and width. Then stage 2, 3, and 4 were spaced out throughout spur development, with stage 5 being the final stage where the flower opened the petals and the adult flower released pollen. So for each of those 5 stages, we collected 5 floral replicates for SEM, and we did the standard SEM sample preparation protocol, which included sample fixation in 3% glyceraldehyde, sample dehydration in ethanol, critical point drying, and then we mounted the SEM samples on SEM specimen stubs and coated it with gold for conductivity. The samples were then ready to be loaded into the SEM to capture SEM images. So the SEM images were captured as follows. We captured SEM images over the spur length, multiple close-up SEM images, and then also we captured images over spur width. Then these multiple images were combined using GIMP, and this high-resolution SEM image were then imported into Fiji, which is image J, and we draw a line over spur length and width, and all the cells encountered by the line was counted. We also did a Crystal-Wallace test to compare for significant differences between the cell number over spur length and width for the mature spurs at species level. Then for cell size, we measured cell size using Fiji. We measured cell length and width. And then we measured 30 randomly collected cells at the base, middle, and tip of the spur. And then we also did a Crystal-Wallace test to test for significant differences in cell size with mature spurs at species level. Then this brings us to results and discussion of the daily spur length growth. So indicated in blue, you can see Nemesia macrocarpa. And I indicated the stages, the 5 developmental stages there, where we sampled on every second day. And then in green, we can see Nemesia barbata. And in red, which also had spur growth 4 days longer, is Nemesia Thromosa, which we sampled on every third day. So from this we can see that the longest spur, Nemesia macrocarpa, also had the fastest spur length growth rate, which were much faster than the other two shorter spurred species. And the results of the ANOVA indicated a significant difference between species and the average growth rate over spur length. Then we did the same for spur length, where we measured spur width for Nemesia macrocarpa over and for Nemesia barbata and Nemesia Thromosa. So in this case, we can see that the widest spur had the fastest spur width growth rate compared to the other two species, which also continued for longer. And then the results of the ANOVA indicated the significant difference between species and the average growth rate of a spur width. And the results from light microscopy informed us that growth rate differences occur, as well as developmental time frame differences occur between the 3 Nemesia species. Now, when we look at cell number over spur length, you can see that for Nemesia barbata, macrocarpa, and Thromosa, the cell numbers stayed relatively constant from stage 1 to 5. Showing us that cell division ceased early in spur development. While when we look at the cell number over spur width, for Nemesia barbata, cell number increased from stage 1 to stage 5. While for the other two species, the cell number stayed relatively constant. The Crystal-Wallace test indicated a significant difference in cell number over spur length and width for mature spurs at species level. The results of cell length, where we measured cell length for Nemesia barbata at the base and in the tip, for Nemesia macrocarpa and Nemesia Thromosa. We can see that the spur of Nemesia macrocarpa is approximately 3.2 times longer than the spur of Nemesia barbata, and that is due to differences in cell length. And we also have to look at cell number over spur length. So for Nemesia macrocarpa in the table, we can see there's more cells over spur length compared to Nemesia barbata, but also the spur of Nemesia macrocarpa is longer than the spur of barbata due to more elongated cells in the middle area of the spur. Therefore, the spur of Nemesia macrocarpa is longer than the spur of Nemesia barbata due to differences in cell number as well as cell size. Then when we compare Nemesia macrocarpa and Nemesia Thromosa, the spur of macrocarpa is approximately 1.5 times longer than the spur of Nemesia Thromosa. However, if we look at the table, we can see that Nemesia macrocarpa and Thromosa had approximately the same cell number over spur length. And therefore, the spur of Nemesia macrocarpa is longer than the spur of Nemesia Thromosa due to cell size only with longer spurs, longer cells occurring over the length of the spur, the middle area of the spur. Then the Critical-Wallace test indicated a highly significant difference between species and the average cell length for mature spurs at species level. Then lastly, when we look at the cell width of the three species at the base, middle, and tip, Mesobarbatum, Macrocarpa, and Thromosa, we can see that the spur of Mesobarbatum, which is 2.5 times wider than the spur of Macrocarpa, and that difference is due to the higher cell number. As you can see in the table, with Mesobarbatum having more cells at the spur width than Macrocarpa. And also, the cells of Macrocarpa at the base, middle, and tip were wider than for Macrocarpa. So both cell number and size contributed to the spur of Mesobarbatum being wider than the spur of Macrocarpa. Then we have the spur of Thromosa, which is approximately 5.6 times wider than the spur of Macrocarpa. And we can see in the table that is also due to increase in cell number, and then much wider cells, as indicated with the red arrows. So the Thromosa is wider than the Macrocarpa due to cell number and cell size. So the Critical-Wallace test indicated a significant difference between species and the average cell width for mature spurs at species level. And this brings us to the discussion of spur length development in Indonesia. In Indonesia, different developmental mechanisms contribute to spur length variation, with the spur length between Macrocarpa and Mesobarbatum being different due to cell number, similar as in Lunaria, where the longest spur had more cells. Then if we look at cell length, spur length variation between Macrocarpa and Thromosa, we can see that the spur length variation is due to cell expansion, similar as in Apologia, where the longest spurs had more elongated cells. Then for spur width development, in Indonesia, similar developmental mechanisms contribute to spur width variation, with cell number as well as cell size contributing to spur width variation between the three species. And it should be mentioned that this is the first study investigating the developmental basis causing spur width variation. And this brings me to my conclusion. This study adds knowledge on nectar spur development in Indonesia for the first time and provides us with information on spur development in different taxa. This study also provides us with a basic model of spur development in Indonesia, which can be complemented by future genetic work, for example, investigating the genes controlling cell divisions before the sepals opened and cell growth after the sepals opened. These are my references and my acknowledgements. I would like to thank my supervisors, Dr. Rios Lubaran, Dr. Marie Jackson, Department of Plant Science, Dr. Leon Duprier for help with statistical analysis, and then the Center for Microscopy for the same analysis. And thank you for listening.
Thank you, Hanley, for an interesting talk. There are lots of questions for you. So the first question is from Glennis, who asks, where are the glandular cells that produce or release the nectar located? Are they inside the spurs?
Sorry, I was just closing my screen. Can Doc just repeat again? Where are the glandular cells that
produce the nectar located?
Okay, so we are still investigating that. For one of my chapters, I looked at nectar rewards in Indonesia. So we couldn't find any stomatal, how can you say, nectar releasing. Sorry. Okay. It would be located, I think, at the base of the spur or at the tip of the spur. The species I investigated did not produce a lot of nectar, But some of the species that produce nectar, the nectar I think forms at the base of the spur, then you can collect it with a microcapillary tube. But we are still in the process of investigating the structures and the method of release of nectar in Indonesia.
Okay, and then Peter Bester asks, why was Thromosa sampled every third day compared to the other two species that were measured every second day?
Okay, that's a good question. For Nemesia's Thromosa, the spur growth development occurred for 13 days compared to Nemesia macrocarpa, for which it occurred for 9 days. So because we wanted to space out the developmental stages throughout spur growth, we sampled for Nemesia macrocarpa and barbata every second day, as that was very nice points spread out throughout development. While for barbata, Thromosa, it was on every 3 days, just because the spur development occurred for longer. So that was the difference between selecting for 2 days compared to 3 days for the different species.
Thank you. Then there's another question on the nectar from Glynis. She asks, do you have any idea whether they produce different volumes of nectar?
I think the different Nemesia flowers will produce different volumes of nectar. I know Emma Ferreira, for example, found some nectar in her floral spurs of Nemesia, but for the flowers I investigated, I couldn't collect any nectar, so they are possibly empty flowers.
And then a final question from Janine Victor. She says, very interesting. Is there variation in mature corolla dimensions? Is corolla development affected at all by environmental factors such as nutrition or light?
Okay. There's definitely variation in the mature corolla. For example, Nemesia Thromosa, much larger flower than Nemesia macrocarpa and barbata, which are very, very small. I think corolla development might be affected by environmental factors. Since the Nemesia barbata flowers we collected from the field, they did not like the environmental changes. So I think it might have, to an extent, some impact on the flower size.
Then there's a final. Oh, sorry, continue.
No, I also noticed that the older the plant became, the smaller the flowers became. That was also quite interesting, that in the beginning the flowers are larger, and as the plant ages, the smaller the flowers become.
That's quite important. All right, so I think Janine then just posted a follow-up question. Within one species, does the size vary for fully grown flowers? So not variation between different species, but within a species.
Yes, they are to some degree there are variation. I saw, especially for Nemesia Thromosa, there's different colors, but the size is not significantly different. It's maybe like a few millimeters that the spur variation differs, but overall, it's a bigger flower than compared to the other species, but there is some degree of variation in flower size in Thromosa, for example.
Okay, and then a question from Nicola Berg. Thanks for an excellent presentation. Do you have any idea why the spurs vary in size?
That would possibly be because of different pollinators. We still need to investigate which spur shape correlate with which type of pollinator, but I would think that different spur shapes would be due to different pollinators. For example, Nemesia Thromosa with the white saccade spur is also not closed with a palate. Insects, for example, beetles can climb in there and collect pollen or nectar rewards, while the spurs of, for example, Nemesia macrocarpa with a long, narrow tubular spur is possibly pollinated by bees, which lands on the flower and then sort of forces into the spur to collect a possible reward. So I hope I answered the question. But I think spur shape is related to pollinators. Yeah.
That would be a likely hypothesis. Excellent, Andy. I think that is all the questions for today. So thank you for your presentation. Thank you to all our presenters for the session, and thank you to everyone for your interesting questions and for attending. Session 1 is still running, so if you are still keen for some talks, you are welcome to hop over and join session 1 for the rest of the day. Thank you very much.